Okay.
Thanks, Robert.
Okay, let's begin.
Okay. Thank you, Danny, for this nice introduction. Thank you. And so, I work currently, I used to work at the U.S. Department of Energy's Oak Ridge National Lab, like for 15 years, as Daniel introduced to me. I currently work at Old Dominion University in Norfolk, Virginia, in the United States. And so my topic you can see here is energy renewal, okay? Isothermal utilization of environmental heat energy with symmetric function. So the keywords are isothermal, right? Okay, if we have a temperature gradient, of course, everybody knows how to use it, right? So keywords isothermal utilization. So my talk, basically, we identify there are another type of energy process. I now currently call it as a type B energy processes. Okay, so we identify they actually have two thermodynamically distinct type of processes naturally occurring on Earth. Okay, we'll give you an example of it. Now, type A, such as ATP synthesis or the classic heat engine process, everybody, of course, are very familiar with. They apparently follow the second law very, very well. Okay, so we already talked about that in the morning as well. Now, the type B process that naturally occurring on Earth, I'm going to give an example of it, exemplified by these thermophilic functions with transmembrane electrostatic localized protons. Okay, we'll talk more about this. Does not necessarily have to be constrained by the second law owing to its special thermodynamic asymmetric function. That's really the key words here, okay? So whenever you have a symmetric process, be careful, don't apply the second law blindly. Yeah, you can do it, but if you do that, you'll miss very important things. That's my message here, okay? So I'm gonna talk about how we discover this type B or identify type B, okay? So now we actually, initially, I was actually not trying to looking for challenge the second law or try to discover, this type B actually was discovered in a sense pretty unexpected. Like about 10 years ago, even like 35 years ago, and when I was a PhD student, we were very interested in the bioenergetics, the Peter Mitchell's theory. It describes how our ATP is made in terms of called the chemi-osmotic theory, right? She got Nobel Prize for it. But there's a lot of debate whether protons are localized, delocalized. Some of the observation cannot be explained by his theory. Although he got Nobel Prize for it, he really made a great contribution of it. But we discovered that his theory is not entirely correct. I'll show you why. So now, the thing is actually everybody understand was it's like a problem that the theory, the textbook bioenergetics theory proposed by Peter Mitchell cannot explain the bioenergetics. It's actually one of them, a very clear example is on the aquaphilic bacteria. Now, these bacteria can grow at pH as high as 10.5, even 11. Okay. Now, so the inside the cell, these people have to measure. Those are the data. It's not serial. These are experimental measurement. Okay, right? Those are the published data. Okay, people measured in the last 30 years. Okay. The inside the pH is 8.2, and now in bacteria, similar to our mitochondria, we can talk about later. There are respiration systems in the membrane. So these are biological membrane. So this is the bacterial cell membrane, okay? So this is the outside, this is inside the cell. The entire cell, this pH is 8.2, so it's normal, just like us. Now, these bacteria have respiration system in natural nodes, well defined, well known. When you use organic materials, oxide oxygen, you pump in electron and pump, so the electron movement is coupled with proton pumping. This is a well-known mechanism. It's no theory, okay? It's a fact. Basically, not exact fact. Everybody knows, okay? Pumping protons out. Now, the proton is supposed to come here drive ATP making. This is called ATP synthesis. It's actually a nanometer turbine. The structure as well is also got Nobel Prize for it after the Professor Paul Boyer at UCLA, actually, he proposed the theory, and also the people resolved the structure of it. Actually, was recently also got Nobel Prize. So this process is well known. It's like a little turbine here, makes a turn, and then actually, when the proton come in here and drive the ATP making from ADP plus the inorganic phosphate, and then when this rotor movings, convert the mechanical energy into your ATP energy. So that's a well-known process, okay? Now, the key here is that you look here. Now, you're supposed to have proton pumping here, right? But look outside, the pH is 10.5. You have very lower proton gradient, is that right? You have less pH proton here than inside, right? Inside the pH is 8.2. You have more proton in the medium, in the bulk, right? So now we use, so these equations are in the textbook based on Peter Mitchell's called the proton module force equation. Now in short, we call it PM8, okay? So the proton module force equation, okay? So that's he defined, that's in the textbook. So is the membrane potential for this bacteria 180, that is water difference across the membrane here, okay? So is, now here is outside positive, here negative. So it's 100, the voltage difference. So we in the bioenergetics, this is called the membrane potential. Now, according to Mitchell's hypothesis is, the proton concentration or the pH, the difference from outside to inside, okay? Now outside 10.5 and you minus that 2.3, right, okay? So this 59 is just a constant, like RT constant, okay, right? And there's a temperature. So if you calculate with the textbook equation, the proton module force only 44 millivolts. How much you required to drive ATP making? You require 478 millivolts. So even use like 3 or 4 protons, you're not enough. So this is like, everybody knows this in the field of bioenergetics. It's like an elephant in a room. The textbook equation cannot explain for 30 years. People have no way to explain this. So like this conjurma, this mystery has been there like for 30 years. Then I'm coming in. So the idea is actually this. So basically the textbook cannot explain the energetic, how you can make ATP. The bacteria grew very, very well, okay? I'll tell you about more. So I'm coming in here, water, right, is can understand as a protonic conductor for these 2 reasons. These are water molecules, so the proton can hop like this, okay, among water molecules. Another way water can act as protonic conductor, water molecule turn a little bit here, that way there, and this, that. And in a sense, proton in liquid water, somewhat behave somewhat similar to the electrons, the Fermi level electrons on the metal, right? Okay, they can kind of, if you have electric field or something, they kind of conduct. Basically, that's what we call the protonic conductor, right? Of course, water does not conduct electrons though, but only to the proton, right? So this is proper to call water as a protonic conductor, of course, the insulator for electrons. Make sure that we understand that, okay, right? Now you understand water is a protonic conductor. Now let's go back to the case here. You have water here, of course, outside the cell, right? You have protonic conductor. Your membrane is an insulator, right? Okay, right? And inside the cell, you have water, protonic conductor. So you have a conductor, insulator, conductor. By definition, you have a protonic capacitor. You got it? Now, so that what happens, the proton pumping across the membrane by the respiration will not go to the bulk, but form this protonic capacitor. This has been never understood or recognized before. So we are the one first, about 10 years ago, recognize this. So this basically is a protonic capacitor, similar to electron capacitor. In that case, act is electron, but in this case, it's protonic, protons, okay? Now, if you Google the words protonic capacitor, it does not find it, because that words I created. If you protonic capacitor, James Lee's name will popping up in Google, okay? So we are the first one to understand that. So now if you understand, there's a layer of proton here, you see that, right? These protons will be just on the liquid water membrane interfaces that are sitting there. They are thermally vibrating. We'll talk most-cited about that. They're going to hit that thing and doing the work, right? Then you calculate this, then as actually the whole thing making sense, you know? So, but you need a modified equation because the textbook equation never talk about it. They do not, they are not aware of there's something like this going on. So what will be the correct proton multiforce equations? So that's really here. Now remember, this is the maturing proton multiforce equation in the textbook. Okay, so it's PMF membranes 2.3 RT. Okay, Faraday constant times the pH difference across membrane. So that's in the textbook. So, this is my modified, revised proton multiforce equations. Now, the first two terms here, you see, these are the same. This term is essentially the same as that, just a different way of writing it. Remember, pH is minus log, okay, of the proton concentration, right? That's right here, okay? So this is on the proton on the p side outside over the proton concentration in the bulk phase, okay? And in the inside. So these two terms are identical to what in the text, but the textbook equation misses this term. So that's a term I added. So here is, is 2.3 log RT, gas constant, environmental temperature. So it's actually, this term is the one here with the layer for on the surface, right? Electrostatic localized, called the localized, I use L here. So the local proton concentration on that layer, protonic capacity, okay? And over the bulk phase in the liquid, the same thing with that one here, okay? And it's this ratio that contributed to so-called, this component actually later we called the localized proton, proton multiforce, okay? So this is very important. I want you to recognize that. Now the question is, yeah, you have a same layer. What the density of this, Yeah, so this is basically we're talking about this layer is a monolayer actually. So you typically will stay within 1 nanometer, not too far, okay? So you have a kind of population density. So I call this called the localized proton density, okay? I describe as this proton little sub L here, okay? So that's just a bulk, just like in water, okay? In the water. So the question is, what is it? Are we able to quantitatively express these things? If we know the quantitative, then you can calculate things. You can calculate energy, right? So the proton multiforce is, if we time this, basically become convert to delta G, right? So in the field of bioenergetics, people use proton multiforce like milliwatts. So that's what we are using. I'm going to actually convert to Gibbs energy later, okay? It's a simulation on that. So my idea of to try to define or to quantitatively express this thing here actually came from here. So we understand the protonic capacity, okay? Now, if protonic capacitor, we should be able to borrow, electric capacity equation to use here, right? That's the whole idea, okay? When we understand capacity, okay? So then we basically borrow the well-known, capacity equation, right? So the concentration of the local proton, I put a 0 here. 0 means like if in a pure water system, okay, just like ideal gas. So let's start from simple, not consider salt. We'll consider salt effect later, okay? Otherwise, human, too much things just like cannot really think through, okay? So if you in idealized liquid water membrane water system, then you will have this. You can apply this if there are membrane potentials. It will be the specific membrane capacitance times your your voltage difference, membrane potential, and divided by the thickness of that layer, about 1 nanometer, and then the Faraday constant. That will be your idealized localized proton concentration defined. Now, so that's good. But in actual biologics, you have a salt, right? We eat a lot of sodium. So in our body, we have plenty of salt, sodium chloride or potassium, there's many things there, okay? So we also further developed a term to call the reduction factor, develop equation to accounting for that. So make the story short, to accounting that effect, we use the real local proton concentration in the presence of those non-proton cation, including this here, M means like cation, could be sodium, potassium, magnesium, all those are in your body or in the outside inside the cell. Okay, they can exchange. Okay, because they are proton, right? They can exchange. If we have sodium here, if you have a large amount of sodium, you can exchange this proton out into the bulk phases. Okay, that's really that term for that. Okay, so when this proton exchange, you have a sodium occupier here. Of course, the density of the proton concentration will reduce, right? So that's I call that is this term basically is a reduction factor. So it's actually the effect is not summation. We figured out it's actually is a product. Like each different you have a sodium exchange with the proton, and you have potassium. Those effects are not additive, but products. Very important. Okay, so that's why this little symbol here means all those terms are product together. Then will be collected accounting for that. So this is very important. Otherwise, the calculation will be all long. Okay, very important. So we divide. This is unique. So you can see already published. Okay, I already published this thing. Now with that, if you know the true concentration of, now you can use this thing to a real world system. So this is the system now. So we apply this, use experimental data. These are people who have done that. So the real experimental data. Okay, so now in the culture medium, all the non-proton cation concentration like sodium is known 300 millimolar in the culture medium. So those are experiment, okay? From it's real facts, no theory, okay? Sodium, potassium known, magnesium, all the evidence known. Okay, so we use the real experimental data for this culture in experiment, and so are known. And another thing is we needed to know in order to calculate exchange equilibrium concentration, we need this called exchange equilibrium constant. Now that thing was not available. So actually took my my PhD student like 5 years to measure these two constants. Okay, a long time. Okay, so that's why we know this thing. Explored this in 2010 or 11. It took like 5 years, and then finally we got this number, and then we can calculate this thing, okay, this reduction factor. So this is the case like in potassium, you see that, right? So it is this fact here. For potassium, the reduction factor in these concentrations that, your potassium, the reduction factor that, and for magnesium, kind of small, equal, close to 1. So the very low concentration, guys, almost to 1. So the total effect is not summation, is time these all together, you have actually very significant reduction factor. Okay, you see that, right? 44 sodium, right? Okay, yeah, so that's for the culture media at pH 10.5. So we have to do each of those pHs, culture pH separately, okay? So because this culture has actually been done, the people culture, scientists culture these bacteria at different pHs, okay? So 10.5 is a number I just showed you, right? Okay. So we did, of course, this bacteria can also grow at 7.5. At that time, the reduction factor is this, okay? So on, okay? Now, so this is the localized proton, like idealized condition. We actually can use the capacity calculator, okay? The membrane potential are measured experimentally, okay? These are experimental data, all those are experimental data, okay? Now, these are calculated from membrane potential, so it's also in the same style from based on experiment. Okay, so these are not like a proposed numbers. Okay, those are the calculated factors. Now use that, calculate the real localized proton concentration. Okay, right? So listed here. Now, when this localized proton concentration is it's here, then the rest of the whole free, you can calculate the local proton motive force. That's the third term in my equation. Okay. Now the classic proton motive force is based on the textbook. The first two terms, and the total proton motive force is two together. That's your total. You see that right? Yeah. Now, so let's plot this out. You're making sense of it. Okay, so I'm plotting this data out. Now you will see it. So this work is already published in the ACS journal. Okay. Now, so this axis is the proton motive force, and that's the cultural medium pHs. Okay, so now the culture able to glue. These are cell doubling times on this axis. So the cell doubling times, like, how long it takes in the cell for making a cell division, making one become two, right? Four become two become four, four become eight, eight become 16, right? The doubling time, okay? So you can see this bacteria. So the doubling time of the minute here, this culture can grow from pH 7.5 all the way pH about 11 here. The doubling time is like below 100 minutes. So like this cell is not slow. Every 2 hours, doubles. Okay, right? Very grew very. This cell actually grew excellent growth. So now when the pH go higher further, and then the doubling time significantly go up. Okay, that was observed. Those experimental observations. So you look at the textbook. Proton motive force equation calculated. The proton motive force, called the classic PMF. You see, now on pH 10.5, you see your proton motive force is about 44. How much required to make ATP for the proton? You need at least 110 or 20. So you have four together, or four together, you were able to meet about 400. You see that, right? That's why, that's why, like, for the last 30 years, nobody able to explain this. These are totally unexplainable with the textbook equation, right? It's the last 30 years sitting there, okay? Now, when you look at that, this also the pattern of the classic proton motive force doesn't make any sense here. Now, then the pH actually go down, right? And then from this point, like pH, from here, you go to 11 and close to 12, you see the proton motive force actually increased. Now, increase your cell doubling time should be getting smaller, not like bending up. You see that the curve should be bending down, right? So not only is absolute value doesn't make any sense with observed true doubling pattern, but also the pattern doesn't match. You see that's why this this thing really cannot explain by the textbook epidemiology theory, okay, per se, okay? So now the reason why it missed the term I discovered. It's the local proton motifs. They are here, and you calculate it, okay? You see that the missed component actually much bigger than the classic one here. So it's not miss a small thing. It's a big component, the layer of proton on the surface, the protonic capacity thing, right? Okay. So the total is these two together. That's your total. So it's way here. Okay. So now you look at this here as pH go up. Yeah, the proton motive force actually go down. The reason is when the pH go high, the proton concentration in the bulk liquid phase go down. So it favors the cation proton exchange. That's why you have this the localized proton concentration go down. So that, of course, the localized PMF component also decrease, right? So you can see here, and that's go down, but still above this minimum required. So you can explain this. That's why this is still able to glue. But when it go down, you see, they have more difficult to double. You see, so now matches. So the total proton modes like this go down, and the doubling time go up means grow slow, right? So now at the first time, this 30 years mystery being solved in terms of bioenergetics, okay? Like why now we cannot explain how this bacteria can grow, okay? They actually have enough energy. You can see that because the total proton motive force is well above that, okay? So that's good. Initially, that's my only purpose. I try to explain, how this thing can grow or how this can make ATP, right? Just all that mystery. But now you look, I want to pay attention to here. Now, this 220 millivolt or 228 or 230 millivolts called the redox potential energy limit. What this came from? Now, in our mitochondria, all the bacteria, they use electron, right? Free electron, okay? Like NADH, okay? Or these organic substrate. So oxide to oxygen. But that redox potential energy, amount of chemical energy available is known. It's about close to about, so the redox potential from, for example, NADH to oxygen, this whole chain is about 1100 millivolts. Okay, so we know that for each electron goes through, they're pumping 5 protons. So 1100 divided by 5, about 220 something, right? That's this chemical energy. So that's your like, even you assuming like 100% energy efficiency, you convert all the food you eat to chemical energy, right? That should be a glass ceiling, right? It should not have a proton motive force, should be higher than that, right? So initially, when this is about 7 or 8 years ago, when I first observed, I thought maybe these my calculations or my equations, I checked everything, was correct. And then I think about exactly where the energy came from. These things well above the energy you put in, your chemical energy, right? Even 100% efficiency, you should know higher than that. So this high is not a little bit, right? A factor 2 here. And so then suddenly the story, my early work in during my college years, like that's like 40 years ago, in 1979 and 1980, I was actually doing a student project to explore the question of whether this thermophilic kind of thermotrophic property or thermotrophic kind of type of life is possible. That means, is it the heat energy in the environment can be used by life to making things, to do work? So actually, if I have time, I will cover that. Actually, I did that. And then I was like, aha, this thing is the thing. Actually, I was looking for this maybe 30, 40 years ago. I kind of put aside because all my professors, when I came to US at Cornell, my old regular professor, they all say, hey, James, environment heat cannot utilize, the thermotrophic things, if you want to do that, we cannot advise you. So I just learned, chemistry, working for Department of Energy, all classic, traditional energy. So I kind of already put that thing aside, all those, very conventional research. And until now, I realized, aha, that's a thermal thing here, because it must be thermal. So I carefully look. Now, so you look at this component, the localized proton motive force, right? You remember this is a local, that one miss. Okay, so it's a concentration of your localized proton versus the one in the bulk phases actually contributed to your free energy. This local proton motive force actually have, as a simple relation become a delta G, right? First, I'm going to explain that. Okay. So in a sense, the protonic motive force for this is just a time, time your Faraday constant. For this one, time Faraday, you cancel that one. That's it. So clear. Okay, right? So because have it's a minus Faraday constant time, your proton motive will become gives free energy. So it's right there. But you look at that. Now, where the heat energy come from? They're hiding right here, RT. So in this audience, I think the R is the Boltzmann constant times Avogadro's number, right? You have a KT term hiding right there. So it's a thermal energy. Your equation tells us here. You see it? It's use this KT term. It's hiding there. In the past, we just kind of oh, gas constant, we just take for granted. That's not really thinking very hard. So I immediately realized this actually, the temperature, remember, inside the cell, outside the cell is the same. There's no temperature gradient here, inside, outside cell, okay? So that's really truly isothermal. So that's why the single T, okay? Yeah, so so that's why we now understand that this really is the energies came from this term called the local proton motive force, are called the local protonic motive force. So where it came from? Now we can also define, we actually find out the entropy change actually shown here for this localized proton can be shown by this equation here. You see, right? You divide by T here, you got that, okay? And that's actually the ratio of the localized proton over proton, the concentration in the bulk phase, in the bulk liquid, okay? Of this system. Basically is this your transmembrane electrostatic local protons versus the bulk phase protons is related to called negative entropy change. Data is L, is mathematically now defined. So now this is very important. So that says that now as long as you have your local protonic capacity behavior that all biological membrane actually has, very important, all biological, I'm talking about not only this bacteria, including our mitochondria, I'm going to cover later, or in the trees, in the algae, or in the trees, in the oceans, in your homes, in everywhere when you have life, you have these things, okay? So what happened here? Well, as long as you have this, so this will be, as long as it's not zero. Now, if zero here, a logarithm of 1 becomes 0, right? Your entropy change to 0. As long as this is non-zero, above 0, you have a negative entropy change. You see that, right? So this is happening. So this really, the first time we mathematically or scientifically discovered the natural system, they have this, they are utilize this natural occurring negative entropy change events. They try to utilize that, basically taking advantage of this entropy effect, able to utilize, attract the energy from a simple vibration of the protons. Okay, they usually will be negative. They will let them. Okay, but they will, if they are in the bulk, no matter they close it down, they're not able to do the work. But if they're electrostatic local surface, you have a high probability of hit that thing, right? That's really this delta G is for. Right? Okay, so it's very clear. It is the they are using environmental heat energy, actually isothermally, because temperature is, you have a single T. There's no difference across memory, okay? So I hope everybody now get it. So this plot, I just convert that to free energy, the proton motive force, convert to, just plot as Gibbs free energy, right? Same thing, right? You see, it's the same pattern, okay? So I just want to make sure that, in biological, bioenergetic is the proton motive force, but in thermodynamic, everybody here in this audience probably more familiar with delta G. So, okay? So you can see here, that's your total delta G. That's a classic proton motive force delta G. That's your local pre-emission free energy. So same thing, okay, right? Okay, I just make sure that's clear. Now, so one thing I want to pay attention, so in that case, how you can break the glass ceiling? Now, I predict from this data, if this is correct, if my interpretation is correct, I would predict the structure. So this is a piece of biological membrane in your mitochondria, okay? It's very similar to one in the bacteria, okay? But this one is like well-defined, everybody actually knows, okay? I expect, so those are the respiration. For example, this system use NADH so electron from here, all the way to here to go to oxygen and pumping protons. Okay, those are the proton pumps. Okay, so I expect the proton pumps, the protein, the exit should be like extended about 1 or 3 nanometers away from the membrane. like this one here, they should sticking out into liquid. And the proton user, the mouth of it, has like the ATP synthesis had to be stay there, okay? Because otherwise, so they now if this side is not sticking out, now if they were pumping against this layer, then you backfire. You see that, right? Okay. So this is my prediction. That's why how can they can break the glass ceiling. So that means this system is asymmetric. Remember, that's keyword I discovered, okay? So these two sides are not the same, okay? If symmetric, you don't have a game. You see the two sides are symmetric. That's really the key here, okay? You see the membrane are symmetric biological. So this came from billions of evolution. You know, these are predicted exactly. These now are the data from cryogenic electron microscopic data. This is true. It's not my cartoon. It's published here in this article. I dig it out, match exactly what I predicted. So I feel very confident what we discovered is real. Okay, so this came from billions of year evolution. It's in you and me. It's in the whole. All organisms on Earth have that. So that's why type 2 process actually is in the past, we just did not know it. We now understand that, okay? So because of time, I'm gonna run a little fast, okay? So this part, the present study, you know, with this transmembrane electro local proton theory, has been better explained the decades-long standing, you know, conundrum, okay, you know, the mystery of ATP synthesis in agricultural bacteria. The calculated PML curve matches excellently well with observed population growth pattern. Okay, that's just that. Now, more importantly, reveals isothermal utilization of heat energy with local proton at the liquid interface to do useful work, driving ATP synthesis. Okay, it's real. Okay. Yes, so it's hydro chemical from active amount of heat. Now that's the thing came from, I'll just talk about, okay, right? And it's the scientifically in this audience, most interesting one is this negative entropy happens, okay? So this first time we really kind of demonstrate this. So this system also happens in our mitochondria, okay? So I'm gonna talk about that. So this in our body, mitochondria, okay, so those are well-resolved structure. You have this inner membrane like this called the crystal, okay? Now the ATP synthesis, actually, they're located on the tip of that, okay? Those are reviewed by cryogenic electron microscopic work. So people in the field cannot explain why they do that, you know? Why are these things localized so neatly on the tip here? We now have a theory here. So for example, why mitochondria develop the crystal? Now you understand, first of all, the formation of crystal creating more membrane in the membrane surface area because this is a protonic capacity, right? The bigger, the more membrane surface area, you have more capacitance, you're able to store more energy, right? That's what they do there, right? That's one thing. But also the story here, you know, you form this kind of structure called a crystal, you know, so you have membrane here, but you look this thing here, there's another effect. For electrostatic, you have this, your proton concentration on the here, because this, you know, this is you have this special shape, okay? It's not round, okay? Crystal. Actually, the concentration is high here. So although the pump, your proton pump, I show actually in mitochondria is here. So they're pumping at the lower concentration part of it, but electrostatically make it even higher. And that's why they're sitting there, they basically utilize the best way to utilize this, you know, heat energy. You see that, right? This again came from billions of years of evolution. They are natural forming, okay? So this is another finding here, okay? This is already published in the Nature's Research Journal Scientific Reports, okay, this work. And also we calculate the exchange effect for this bacteria, for mitochondria in our body, okay? That factor is, you know, the reduction factor. So we calculated from there, we calculated the mitochondria without crystal is here. With crystal, you have much more proton, local proton per mitochondria. That's evolutionarily make that crystal formation have advantage, although you don't have to. You know, some of the bacteria may not have, but the more you have more protons, local proton, right? So you can use the heat energy more, you see? So evolutionarily, they're making sense. So that's another piece. So also in this article, we discussed that. And more important, we calculate the energy for mitochondria. Okay, so I'm gonna, because of time, the calculation is similar. Okay, so I'll calculate, in this case, I'll calculate the data S, okay, you know, with the membrane potential. Those are experimental, okay? And these are the calculated data from the real experiment. And you see the first time you numerically demonstrate data is a negative number, okay? First time. So it's this case is locked, okay? It's locked, it's a thermotrophic feature. And these are the data G for your localized component. This is the classic, the total is this. That's your, how much is needed for making ATP. That's your chemical limit, because all the chemical energy, see, the requirement is when, you know, higher than the chemical energy. Chemical energy cannot explain ATP making your body. Okay, that's basically what it is. So that's how the plot is, okay? So that's your classic in your mitochondria from membrane potential in your body can from 60 to 200 milliwatts. And so that's your classic proton free energy. That's how much you require. So this, the classic calculation really cannot explain how a mitochondria make ATPs. Now the local proton modes here way above that. That's your total. And that's your required proton motive force. So our efficiency about 50 or 60%, you know, it's so the system, they grab a significant heat energy. They use, you know, they use very freely. They use very generously. They waste a lot of heat. It's also, so second law in that case applies very well. So don't say second law is wrong. We are not checking second law. Yeah, so second law only when in a free system, yeah, you do. But for the membrane, you have a two-dimensional thing, you have asymmetric, be careful. So second law does not apply, okay? So just, that's type of B, okay? Yeah, so I already showed you this. So this is, you know, a symmetric thing. I just revisited, okay? I showed you before. And how this energy is exactly used. Remember that ATP is here? This is structure is resolved. So let's look, you know, where this structure is, proton how you use, you know. So remember that thing, okay? So this is the resolved structure. You see from the science paper, okay, right? So this is structure. So that's a liquid phase, okay? Your water here. So the proton actually come right here and drive this turbine, making this more, and you drive ATP making, okay? Where the proton is getting, you see, that's your liquid phase. Remember the local proton is right within this layer, and there are thermal vibration. Remember you have a KT, right? If there's here, of course the protons lateral, they don't have I, they bump in and out. But if there, they're gonna hit the thing, right? When they hit it, they will turn them out, turn these molecular turbines, make ATP, okay? And then, so that can summarize this. That's how that heat is utilized, okay? So ATP theory, with its newly formulated proton motive force equation, cannot fully elucidate energetics for so-called oxidative phosphate in mitochondria, okay? You know, for the whole membrane potential. This study has not revealed mitochondria can isothermally utilize environmental heat energy with ambient temperature, in that case 37 C, okay, of the thermokinetic energy of the proton, okay, of the local proton to perform useful work, drive synthesis ATP, convert heat energy, part of heat energy. Of course, some of the energy came from chemical, okay, chemical energy as well. So local proton motive dominate the TMF and the mitochondria obtain as much as 70% of the free energy from this process. So mitochondria part organ, including us, are not only chemotrophic. Remember, we need to eat food. We are chemotrophic, okay? But we also have a thermotrophic feature. Isothermally utilize the environment heat energy, okay, to help ATP making. And here is my take-home messages, okay? I'm going to probably stop here because of time, okay? I can run you through this, okay? So basically what this thing here is, the key message is that this study review as a thermotrack feature that biological membrane system can isothermally utilize environmental heat energy associated with ambient temperature, thermal motion kinetic energy of localized proton to perform useful work dry ATP making. And the local protonic free energy from isothermal environmental heat utilization with TELP is expressed in this equation I showed earlier. It's not quite clear. There are two thermodynamic distinct type of energetic process naturally occurring on Earth. Type A is everybody familiar with. Type B, as example in my talk here, does not necessarily have to be constrained by the second law, owing to its special thermodynamic and spatial asymmetric function, as I show in the membrane, associated with the natural entropy change. So thank you.
Okay, thank you, James. So let's begin with questions. I'm going to check first on participants. Okay, there are a few hands, I think. Okay, Dr. Solomon, you're up again.
Okay, second time at bat. Basically, my question is, this is great results. And as you said, why haven't people discovered it before? I'd like to get your opinion as to why this happens. You know, what goes on in your mind, it might not be going on in their minds, or constraints that they have that you don't have. But at any rate, my main question is, has any of this been applied by the neuro people to create better models of neural transmission and stuff like that? Because my neuro group in this area has found it to be incredibly incorrect and lackluster and old time ways of looking at neural structures. They don't even get into the sodium, potassium, calcium, magnesium. They don't get into any of that stuff as well as they should. And I was wondering, at your level, you have a lot of stuff there that could be used in better understanding neural firings and transmission. And do you or any of your colleagues or follow-on people have used this in their model?
Yes, thank you, Robert, for your question. Yes, indeed. I already have a publication on applying this finding to the neuron. Actually, so friend, everybody drinking water, okay? Because water protonic conductor. Robert, the signal from your brain to your leg is one neuron inside the liquid. In our brain, in our body, there's no electronic circuit, because the shortest, the longest circuit is the mitochondria, like the, like 30 nanometer cannot form a circuit. What happened? So apply this one here is the water in the neuron axon are very long, right? So like more than a meter from your head is protonic signal. So I actually already have publication just published about a year ago in neurophysiology. Actually, I already, Robert, I can send the article for if you can send me an email. So I will be able to forward my article. Yes, in fact, from my work, although it's not covered here, our brain actually working on a protonic circuit. The neuron, all the neurons together, right? It's the actual potential is actual protonic signal, because it's the membrane potential. Remember, is from the protonic, capacitance signal, right? The protonic capacitor, right? Okay. So in a sense, we now understand our brain and our neural system. Yes, all the neural theory, because they always talk about sodium diffusion, they cannot explain how the conduction of a signal, they're too slow. The diffusion too slow. Diffusion of sodium is like very slow, right? Cannot explain, how our pace cell in the heart works. And cannot explain, I'm talking very fast, my signal to my hand or to my muscle, okay, right? For diffusion, no way, but protonic conduction is very fast. That's why, yes, this Robert, this has already applied into neural physiology. So I have, already published that. Yeah, thank you.
Okay, let's move on.
Just a real quick comment back, if I may.
Okay, very quickly.
What we're getting out, what also is ignored, is the fact that the water molecules are rotating. So you
Yes.
get rotating water molecules, you obviously get magnetic moments and all sorts of coupling effects and whatever.
Yes.
And our work has shown that when you have high sodium content in a human being, very high sodium content via salty diet or whatever, that this definitely affects the nature of the rotating water molecules, and it has impact on neurological function. But more of that perhaps some other time.
Thank you.
Thank you.
Okay, Justin Coven.
Hi, James.
Yeah.
My question in regards to, you were indicating that there's there's pulling of heat from the environment. So, you know, I don't know if that's an assumption or if you've had measurements of that, of actually seeing the heat coming in from the environment as this process is happening.
So, so the heat is right here, Justin. You see the RT, right? As I pointed out, R is the KT, Boltzmann constant KT times your Avogadro's number, Na, right? So it's right there. Exactly, explicitly tell you that. That's the energy you're getting from the environment. Is the KT is well known is the kinetic energy of the in this case proton, thermal random thermal chaotic thermal motion. But when you the proton dancing around the mouth of that ATP sensor, you're going to hit the things, how to harvest it. so it's here.
Yeah. So you know, there's a question of whether it's you know, as was mentioned by Thomas Malone earlier.
Yes.
Is that energy coming from 0 point, or is it coming from external and you know, heat from the environment? So you're showing that there's something that's occurring here, and it matches the patterns that are you know, actually seen in the cells, you know, from a capacitor perspective. So there was some, you know, really solid stuff that seemed like you actually had experimental data behind it. But the question is, do you have something experimentally that's actually showing that there's some heat drop from the environment? You know, now it could be you're just talking, well, it's just normal diffusion of heat from the environment. And
yeah, so very good question. So this I'll go back to the when I was a student. You see the year of 79, 82. At that time, you know, this me, it's 40 years ago when I was a student. I have an idea. I want to explore whether, you know, the thermotrophic type of metabolism is possible. So we actually did experiment with the heat absorption of thermophilic bacteria. In this case, actually is this bacteria, this methanol. Okay, we enrich it. You know, that's like we done 40 years ago, students. That's self is a great achievement. And then we put this in a reactor. We measure like this your culture with it's acetate medium. They fermented to so the organism, okay, and they metabolize making methane. They actually theoretically absorb heat. Okay, so but we want to test it, make sure they are really true as you said. Okay, we predict they do. So this is control. So this experiment done. So this like a system, you have a reactor here, you control, you compare the temperature difference. So this whole thing is in a thermal box. Okay, all right. So you control actually. We measure the heat difference. Yes, they are temperature gradients. So this in Chinese. So just this is a temperature difference. Yeah, they are absorb heat. So the temperature on your treatment is slightly lower, 0.1 C centigrade lower than your control consistently. We repeat 4 times. So we do observe the organism, in this case, in the total system, actually absorb heat from the environment. So thank you, Justin.
okay.
Any other questions?
This actually is this early exploration. Actually, when this big curve came up above the chemical energy, that's in my bell. You know, the sign I'm looking for 40 years ago said, James, your sign is right in front of you. Why don't you recognize it? And then, oh my gosh, this I'm looking for like 40 years ago finally came to me. I selected from the data. Okay, thank you.
Thank you. Garrett.
So this question is really perhaps more due to my ignorance of these types of chemical processes.
Mhm.
But I see that your reaction takes more than a thermal energy. That
Yeah.
really proved quite clearly.
Mhm.
My question is, where does that energy come from? Because in this chemical soup, there are many sources for the energy. So can the energy be transferred from some other chemical in the process to the particular chemical that you're measuring?
Yes. So, Professor Mao, that's a very good question. So in these bacterial metabolism, the net actually is absorbed heat, but in our body, we have other process, like when we eat a lot of food, we have oxygen, right? So this one does not use oxygen, okay? So they are, kind of fermentation, right? They release less heat energy. So the total actually is absorbing. But in our body, we actually, we still chemotroph, we eat food, we oxide, we also, pumping protons, we release a lot of heat. So it's a mixture of the chemotroph. Don't forget chemotroph. We are not thermotroph, pure thermotroph, okay? We are a mixture of the chemotroph, we release heat, but in the last step of the proton on the surface, grab a significant energy back. So that's really the reason why, for one audience say why people did not discover, because it's difficult, because it's a mixture. You have a chemotroph, you release a lot of heat. That's why I maintain our body temperature 37, right? Of course, we are close. Sometimes we try to take in hot shower, make it warm. But in overall, actually, the chemical energy from oxidation of food, we still release a lot of heat energy, but we grab them back. That's why our efficiency, our body's metabolism is so high. It's even related to the equation of why we are 37. Why we are so difficult to lose weight, because the energy efficiency just not like the second law apply, like even 60% or 50% efficiency. Yes, the chemical part, yes, but the last part grab a significant back. So the total energy is like closer to like 90% or something. You know what I'm saying? So, but still you still produce heat. So we are actually in human, because we are oxidative, we are hot organisms. We make our body hot. We actually have a lot of heat source from our oxidative process, like when we pumping protons, when electron moves, and also the chemical reaction, provide vibrations, like in the medium, we produce heat. So that's why that's why people, was wondering why biological systems sometimes have such a high efficiency, but we try to still explain by the classic way of explaining. But that's why the debate, so now we understand, oh, wow, you actually have two things going on. Okay. Yes, thank you.
Right. Well, James, to follow up on that, I mean, I would assume metabolic studies have been pretty rigorous over the last 100 years to try to balance heat production, waste production, CO2, water production by organisms, human beings and other animals, against the intake of energy. And if 70% of energy is actually coming from the thermal bath, how does that square in your mind with metabolic studies that have been done over the last century?
Now, if you measure the total as Garrett just the question is, if you try to measure the total, you still have the total process still release heat. So that because look, the second law is overall like a mythical. If you don't know it, I was thinking that second law still applies very well, okay, right? So what happened was the heat release process actually, the thermal efficiency, the energy efficiency is like about 50 or 60, even like 30, percent, depending on status, okay, depending on what status. But the proton things fetch a lot of come back. So your total still is not like this bacteria. This bacteria is special, okay, because they are endolopic, okay? And that's why in this bacteria, you can measure. They are the net heat absorbed, okay? But in human, we are net heat releasing. So that's why this thing is not apparent, okay? You got it, right? Yeah, thank you.
Okay, I think we're just about out of time. So again, James, thanks very much for a really provocative and interesting talk. At this point, I'm going to turn it back over to Garrett for the next speaker, I believe.
Okay, thanks.
Right.
So I'm going to stop sharing. Okay, good.